cytokines. Cytokines are really important because they're the messengers. They're the way that these blind cells can communicate. They use chemical messages. But they're also a little difficult to study because there's so much going on. I mean, you can have several different kinds of effects. They can have several different functions depending on where in the body they are and what types of cells they come into contact with. So being able to become an expert on cytokines in one lecture is impossible. I don't know enough about cytokines to really be able to give an in-depth lecture on it. And I was looking through, and there's large chunks of this information in this chapter, especially on any of the signal transduction pathways and that sort of thing, that really aren't necessary for your understanding of this topic. I mean, even if you're going into medicine, if you're going into pharmacy, even if you're wanting to go into immunology, this is kind of a freshman level immunology course, not freshman level because it's split with graduate students. It's still difficult, as you know, but there are chunks of it that I just don't think you need to have to memorize for an exam in this class. If you need to know exactly which cytokine does what, you're going to look it up. So we're going to go through the beginning of this chapter and then jump over kind of to the end of the chapter and leave a large chunk of it out, which will help you study for your exam. Now, there's some properties of cytokines that are interesting. So read through that first section. You will also hear them called interleukins. They're all kind of named IL, IL-1, IL-2, etc., which makes it a little easier to keep up with what they are. And they're really just chemical messengers that can carry out different kinds of effects. There's an autocrine effect, there are paracrine effects, and then there are endocrine effects. So let's see, the analogy that I like to use for this is, let's say you're a gassy person and you have a big bowl of chili the night before, but you got a head cold and your sinuses are all stopped up. So you pass gas in Walmart and you can't smell it because your sinuses are stopped up. You can't smell it. So that is not going to affect you. It's only going to affect the people around you. Okay. So autocrine would affect you. That's when you produce the cytokine and it has an effect on you. Now, you can also take the paracrine action, and that's where the people around you scream, grab their nose, and run off. That's usually within a fairly close proximity to the secreting cell. But then the endocrine effect is when it travels for a long distance. So you're walking when you pass the gas, and it gets picked up by currents of air in the Walmart, and they get carried over to an aisle or two away. Then that would be an endocrine effect. It's affecting people 2 aisles away. So that's kind of how I look at that. I know it's a strange analogy, but it's one way of helping you remember the concepts there. Now, another one of the reasons that it's hard to study these types of chemical messages is that they can have multiple different effector functions or effector mechanisms. Some of them, like IL-4, for example, is pleiotropic. It depends on what cell it comes into contact with as to whether or not it's going to cause them to go through activation, proliferation, and differentiation, or if it's just going to cause them to proliferate. But the point is it can affect many different cells. It can affect B cells, regular thymocytes, or even mast cells. It can cause a dramatic increase in the number of mast cells. Then there's redundancy. Several different cytokines or interleukins can cause the same effect on the same cell. So IL-2, IL-4, and IL-5 all cause B cells to go through proliferation. So if you're trying to set up experiments to study these, you knock out one and another one takes over. In many cases, you don't even see the differences in the knockout mice that have one of these knocked out. It may take multiple knockouts to actually get an effect. Some of them work synergistically together, so there's synergy. So if you get both IL-4 and IL-5 secreted, then you get a class switch occurring in B cells. But it takes both of them being secreted in the same general proximity to induce this class switch to occur. And then there's antagonism. You can get interferon gamma can block the activity of interleukin 4 on B cells. So some gamma interferons can stop the progressive, I guess you would say, proliferation of B cells or certain cells in the body. That's why they use it some in different types of hepatitis and cancer treatments. Is you can use this gamma interferon. The cancer cells are affected by the virus, and the virus causes these cells to try to proliferate very rapidly. So if you're using gamma interferon, it blocks the chemical messengers induced by the virus, IL-4, and it slows down the proliferation of those cells that are being stimulated. Okay. So, if someone has hepatitis, they may have to go on interferon treatments, and in some cases, those interferon treatments can go or be required several times over the course of their lifespan, because hepatitis is like forever. You don't ever get rid of it. All right. One last thing right at the beginning of this chapter. It's on page 305, and there's an ELISA assay for a cytokine. They always use that sandwich ELISA test for cytokines because they have the antigens and antibodies, and they have multiple antibodies that can bind to single epitope cytokines, and they're just better at giving you a positive result if you use the sandwich ELISA. It's a little more quantitative as to the number of cytokines that are there, and the assays are pretty well worked out. You can buy kits to do the sandwich ELISA test, so you could take blood serum from a patient and check them for a particular cytokine, like if you think that interleukin 12 is being dramatically increased in production and may lead to toxicity in a patient, then you can run one of these sandwich ELISA tests. The tests run about $500 a set, but you can run 96 samples on a plate. So if you were trying to run this in a hospital situation for $500, you could probably run 85 or so different patients. If you wanted to triple check your results even, or double check your results, you could run at least 40 patients on there. So that's why a lot of times these tests are segregated or regulated. Not regulated. They're sent out to reference laboratories. It's because the reference laboratory is getting them from all these different hospitals, and they can run all of them at one time, where a single hospital may only need to run this test once in a while. It would be cheaper if they could run it through a hospital situation. Now, cytokines do belong to 4 families or groups of chemical messengers, but I'm not going to make you learn that. Repeat, you don't have to learn the 4 different types. They do have numerous different biological functions. You can read over that or look at table 12-1. Both are kind of good, but I'm not going to expect you to memorize which interleukin affects eosinophil activation and generation. Cytokine receptors, you understand signal transduction. You've covered enough of that. Not only in this class, but in any cell biology class you've ever had. So there are different superfamilies, and yes, during a regular semester, I might spend a whole lecture talking about cytokines, especially concentrating on the receptors. But I just don't think it's going to be something that we can cover in this short period of time. So please, just glance over that and make sure you get the gist of what's going on for any future studies that you're doing. But I wouldn't concentrate on that or the cytokine receptors initiating signaling. The next topic that I'm going to actually spend any time on, I'm going to talk about cytokine antagonists. That'll be a fairly short clip, but an important one, because I think this does apply to pharmacy and to medicine. So I'll spend a few minutes talking about that, and then we'll move on to the next clips.